Hi, everyone. I'm Mike Shagru, and I'd like to thank the CNS for having me here today to talk about connectomics and machine learning and how it impacts brain surgery. And we think will change a lot of the ways we do supratentorial intra-axial brain tumors and other supratentorial cases. As a disclosure, I'm the Chief Medical Officer for Omniscient 08T. So for those of you who are not familiar with the term connectomics, it's a topic that I've been involved with for many years now. And basically, it really refers to the term big genomics and pulls the concept of using big data to understand aspects of biology. In this case, it's connections of the brain, both functional and structural. It's mostly MRI-based. There are ways to do things with EEGs and other forms of connectomics. It's really more of a concept, but basically, most clinical applications of connectomics are aimed at fMRI and diffusion tensor imaging or similar modalities. Where it really becomes a valuable adjunct in our armamentarium is when we begin to think about cases like gliomas. Fundamentally, we thought about gliomas for a long time in terms of being a discrete mass with some satellite cells around there. But in reality, we know that in some places in the tumor, it's not just tumor and brain, it's sometimes tumor and brain in the same place. Sometimes there's functional areas in the tumor. And furthermore, tumors can often reorganize the brain as it's becoming increasingly recognized. So when we start thinking about bringing connectivity into the picture, what we're really thinking about is how do we figure out what's important and what are our goals of the surgery? And this is especially apt given that in a glioma, we do not see the majority of the relevant anatomy, the fibers and functional areas around the brain. So when we start thinking about cognitive workflow, going from left to right in this slide, we're thinking about how do we take a very complex insular glioma like this, figure out its anatomy from a tractographic and functional standpoint, as well as the cortical origins, to identify brain networks and turn this into an actionable plan that we can do in the operating room to achieve a good oncofunctional balance, removing as much tumor as possible while not making inappropriate trades. Note that thinking about connectomics and integrating it into your brain surgery does not mean that you never ever cut out a functional area. You always have to cut the brain. And it also doesn't mean that you never leave tumor behind. It's really comes down to making the decision about where that balance lies based on facts, not based on arbitrary mental images of what you think is going on, but basically based on patient-specific anatomy. And fMRI and diffusion tensor imaging are not new. Scanners have been able to do them for a long time, and most scanners in the world will be able to do them. It is possible to do them even in developing countries. And the technology and the algorithms were really first invented in the 1990s. They've been described extensively in at least 550,000 papers, and pretty much everyone in neurosurgery has heard of these concepts and is vaguely familiar with them. So given that they're so established, why don't we do them on everyone who walks in the door? Why is this not part of standard practice? It didn't take this long for us to incorporate structural T1 scans and CT scans into the workflow. And there's a lot of reasons for this. The biggest one that I encountered when I first started doing research in the area is that the software solutions are limited. And as a result, it's difficult to acquire the images. You have to take the images, get them to an expert who knows how to string together a pipeline of open source software usually, or knows how to work with existing FDA-approved solutions, and often requires expertise, sometimes extensive amount of expertise, particularly if you're doing advanced stuff. And because it's so difficult that even at the most advanced hospitals, high-level quaternary neurosurgery centers at university hospitals and world-class institutions, then what happens is that it gets done rarely. And when it's done rarely, we don't really learn very much on what to do with the information. We don't know how to make the decisions because we don't get it. Because we don't know how to make decisions, we don't get it. And so this becomes a kind of a vicious cycle. One of the things is this: it's clear that these kind of imaging modalities can really make impressive connectivity-based images. So when we look at an image like this, diffusion tensor imaging, it's very beautiful, and this makes good wallpaper, good posters. It's eye-popping at showrooms or at meetings. But one of the problems is that because of the extreme complexity of the number of connections and our limited understanding in the field of what those connections are doing, it often is very challenging to really leverage everything that's in here. So while most people recognize that the blue tract at about 6 o'clock on this diagram is the corticospinal tract, very few people know what all the other pathways are doing, particularly some of the ones in the periphery. Now, it's important to note when we think through these images, what needs to really be done. So what you're looking at here is a resting state fMRI. This is what the raw acquisition. So this does not look very impressive, and the raw data by itself, unprocessed, isn't very impressive. It doesn't tell us much information. In fact, it's a relatively low resolution image. But underlying it is a bold signal that has a very rich data. So this is the bold signal at 1 voxel in the brain. And you can see that it's telling you when that area is activated and when it's not activated. But when we start thinking about that, let's say we were to look at the data at every single voxel on that MRI. It's an extremely high dimensional data set with hundreds of thousands of data points to talk about. And so it's very challenging to be able to make sense of this. In fact, we really need machine learning and powerful computers to help this transform this into something that we as humans can act upon. And when you get into machine learning, you quickly learn that one of the first steps in taking any data set that's extremely high dimensional is dimensionality reduction, which is utilizing a method of some kind, usually multiple methods, to reduce the number of variables that you're studying from somewhere in the hundreds of thousands or millions to a more manageable number. This is because most machine learning and statistical algorithms don't work as well or at all on extremely high dimensional data. And so there's a step in dimensionality reduction that almost everyone is familiar with. It's probably one of the better ones because instead of being based on an unstructured data analysis, it's based on rational boundaries inside of neuroscience, and it's linked to something we care about, which is function. And that's the concept of parcellation. What you're seeing here is what parcellation in action, it basically means to take different areas of the cortex and subdivide them based on some property. So for example, Brodmann did one of the first parcellation schemes, and he did that based on cytoarchitecture. The parcellation I'm showing here is the one done by the Human Connectome project, who did it based on functional MRI. But the bottom line is that by dividing areas into functionally distinct regions, where the boundaries of those regions tell us functions that are different from each other, we then have the ability to look into the data and cut this down into an understandable number of variables. Now, once you parcellated the cortex, let's say you want to look at either fMRI or DTI, you now have a lot of power on this. And there's a couple of benefits to doing this that are very helpful. In part, first we can look at how every area has a bold signal, that how much it correlates with every other area. And this is a term called functional connectivity. And where it really becomes powerful is when we begin to know things about what these areas are doing. And so again, you can generate a set of correlations, and ultimately, when we have some prior knowledge, for example, we know what area 44 on the left side is probably involved in, it's language, or what area 4 is, it's a motor area, we can then begin to infer things from the data that make sense. And functional connectivity and other related connectomic features provide us a lot of interesting insights into the function of large-scale human brain networks. We know that areas that fire together tend to perform common types of functions, and that areas that fire together are usually wired together, meaning there's usually a direct physical connection that can be demonstrated on diffusion tensor imaging or other modalities. And beyond just being a scientific principle, this has really practical and pretty profound implications. when you start to look at the literature on functional connectivity, which has been a concept for over 20 years. So for example, if you look at mental illnesses like depression, not only do we find that they almost universally have abnormal functional connectivity, meaning that areas, parcellations, for example, that are generally supposed to be in sync or out of sync begin to have functional connectivity or correlations that are not expected to happen in normal individuals. But more importantly, this has important implications. So in this paper in Nature Medicine, this group demonstrated that using functional connectivity, you could divide depression into at least 4 different diseases that had different symptoms and different responses to treatment, suggesting these are different biologies. And so functional connectivity is generally expected to become increasingly important in the management of brain disease. And it's only really achievable with computers. We didn't know this when we were just staring at fMRI waveforms. It's a statistical argument. So several years ago, after this paper was published in Nature in 2016 by the Human Connectome Project, where they reclassified a parcellation scheme of the human cortex based on task-based fMRI, probably about 20 different types of fMRI, and then cortical thickness and degree of myelination, and then took all of the consensus or a consensus of all of the different ways people had parcellated the cortex and found which ones best fit the data. They were able to generate 180 unique areas per hemisphere that behave different from each other. Now, in some cases, for example, if you see like area 4 or area 44, these are Brodmann areas, and areas like 8C are subdivisions of Brodmann's area. So Brodmann was basically right, but it was a little bit oversimplification. These areas actually had subcomponents. But if you look at things like the temporal and parietal lobe, these areas were renamed towards a different parcellation scheme. And as you can see in the medial occipital lobe, this is the Hubel and Wiesel nomenclature, the V1 to V4, which was a really better assessment of the neocortex. Now, one of the things that made us interested in this is several things. Number one is that you see some very interesting findings when you stare at this. For example, the opercular cortex on the undersurface has distinct areas from the outer surface. And there are areas that sit in sulci, that sit in the cingulate sulcus, and other areas that we don't think very much that are actually quite important. Meaning that the brain really doesn't care what our definition of gyri and sulci are. It organizes function the way that it feels it should be organized. More importantly, this allows us to have an alphabet for describing function in the brain. We've really wanted one for a long time. People have used gyrus names, but of course we all know that gyri are not very consistent, and particularly in certain areas. And furthermore, they're not really the unit of function in the brain. Gyri can have multiple different functions in different parts. In some cases, there's areas that cross gyri from a functional standpoint. So this is not really the best alphabet. And having an alphabet for describing brain connectivity is essential. We need to have a common way that we describe parts of the brain that are used the same way, no matter what country you're in or who you train with, so that we can iteratively improve and get better. We need to describe brain anatomy the same way we describe skull-based anatomy or any other type of anatomy. You need a scheme to do this. And the neocortex, being the most complicated thing we know of, requires a fairly complicated scheme. However, the nice thing about the HCP scheme is that it's learnable. There's familiar terms, and there's a fixed number of terms that are reasonable. So many people are likely familiar, but if you're not, we spent quite a bit of effort to document the anatomy of this scheme in extreme amounts of detail. And our goal was to really have a comprehensive atlas of where everything in the brain was communicating across numerous individuals using different techniques. But while that's very useful and interesting, the feedback that I immediately got was that while this is interesting, how do we apply this in the real world? That's a really interesting question, because it's great to know brain anatomy or tract anatomy, but if you have no way to act on this in the real world in a meaningful and reproducible fashion, then it's just, you know, an academic exercise. And so in order to really solve that question, the first step you had to be able to do is to atlas a brain. By that I mean to take an individual, that, for example, one you're planning on operating on or treating, and to define where those parcellations, those brain regions are. So you can see here, this is a technique for atlasing a brain called an affine transformation. And in short, what you do is you take a standardized atlas that assigns different places in space to parcellations, and you warp it onto the brain that you're going to be working with. Now, this does a reasonably good job, but as you can see, there's some areas, particularly over here on the left side on the opercular region, and on the right side in some of the insular regions, where there's areas that don't line up perfectly. However, when you take the type of patients that we see in neurosurgery, patients with brains like this, these techniques fail miserably. And the reason is twofold. Number one, because of the distortion of the brain, replacement by tumor, et cetera, the assumptions of an affine technique like atlasing that I showed earlier are violated. For example, we know that some of that area is probably doing a very different thing, if anything at all, maybe all tumor in some cases. But also, we're ignoring the fact that some of these areas may have reorganized or been pushed out of the way. And so we really have no way to handle a brain like this. So in order to be able to tackle the complex problem of assigning regions of the brain to parcellations when the brain is shaped abnormally from a brain tumor or surgery, we developed an advanced artificial intelligence technique called reparcellation. Reparcellation utilizes machine learning to look at a brain, take a generic atlas, and adjust the boundaries of individual parcellations, such as Broca's area here, by looking around the area, looking at the structural connections on diffusion tensor imaging, and identifying that this voxel should be part of 44 based on the fact that it's connected to language areas, and that this voxel should not, based on the fact that it has other connections. But also, it acknowledges the fact that the initial boundary may have been wrong. And so, for example, this voxel here also gets assigned to Broca's area, and it moves the boundary, assigning a new parcellation region. This is very helpful in brain tumors when the initial parcellation is in the middle of a tumor. So, for example, this voxel here doesn't get assigned because it has no connections. It's just in the tumor. And this voxel, which was not initially assigned to that parcellation, is outside of the boundary. And so now you see we get a new adjusted boundary that's outside of the tumor. And this allows us to parcellate any brain regardless of shape distortion. So one of the benefits of using a technique like reparcellation is that because it does not make assumptions about the shape of the brain, areas are defined by their connections, not by where they're positioned in spaces. And this makes it more robust to shape distortion. More importantly, it allows us to parcellate any patient to figure out where areas are and what the anatomy and connections are based on the anatomy of the connectome. And what we found is that this model performs remarkably well. For example, in this case, which is one of the worst I could find in my library of cases over the years, you can see a patient who's had a frontal lobectomy for glioblastoma in the past. And unfortunately, this tumor has recurred. You can look in the axial on the upper left and see that the insula is shifted anteriorly because, again, it's going to shift into the cavity. And despite this, it's still able to recognize the basal ganglia and the putamen. It's also able to recognize that certain parcellations don't exist, so it can tell you that they're not there because it can't match any tracks to those. It can tell you also areas inside and around the tumor. And while we didn't operate on this patient, it still shows that even on the absolute most extreme case we could find in our set, it still did fairly well. And we would not expect an atlasing technique to perform anywhere near this well. So it really shows the power of utilizing an intelligent machine learning model to find brain network location. So as we see when we look at tractography, and this is constrained spherical deconvolution, it shows crossing fibers better than DTI. It's a next generation technique. We're able to show that there are areas in the SMA system in this middle of this low-grade glioma, and that tracks are running through there. Now, what we don't know is that those tracks are essential, but we do know that those are plausible locations for tracks leaving the SMA. And so the first question of whether or not there's essential is a scientific question, but we now have the ability to begin to tackle this question. We know that there are things that appear to be white matter pathways running through the tumor. So one of the questions we had when we asked, how do we validate this? How do we know this atlas is accurate? And that's a very interesting question. When we thought about this, we determined that no, it's actually a different atlas. It's very similar. It's somewhere between 90 to 93 to 97% the same. A certain number of voxels get reclustered, but it really is its own atlas because it's doing the concept of atlasing using the HCP nomenclature, but doing it as an atlas of circuits. And it's really challenging when we start thinking about gliomas. Are the areas the same? If a patient has a very diminished area 44 or no area 44, but still is able to talk, what is doing that function? And this is, again, a scientific question that hasn't been answered yet. But what the key thing about it is that we now have the ability to begin to ask that question and answer it using data. And furthermore, to begin to look into the black box and start to see what's going on and making decisions based on improved understanding of what the anatomy of the patient we're operating on is. Now, if you look at how to apply connectomics, and I think one of the first things you have to think about is, what are you trying to accomplish here? So just knowing a pathway is there is not very useful if you cut across it or you overestimate its importance and get nowhere near it. And so there's a balance, just like everything in surgery, between extent of resection and functional outcome. So what is tractography or connectomics useful for? Well, what it's useful for is beginning to be able to make decisions based on data. And particularly when you start looking at gliomas, the fundamental problem that we face in this tumor is a lack of anatomy. We don't see what's going on. We don't see what's in the tumor. And oftentimes we don't have a well-formed framework how to think about the case we go into. So many people just go in and suck the center of the tumor out with a CUSA, and then are sometimes surprised when things don't go the way they expect. What connectomics allows you to do is to begin to think about dividing the brain and separating the tumor against those networks in a way that preserves the networks, keeping them connected to their destination while removing parts of the brain that we think are tolerant to that kind of resection and expendable. So while all parts of the brain are doing something, we of course know that some parts are less tolerated than others. And connectomics starts to give us an understanding to how do we iteratively improve our surgeries to begin to minimize the number of times that we're surprised by what happens. While you may look at a patient with a motor cortex tumor who is paralyzed and say that there's nothing we can do about this, the issue is that it's important to know that that's for sure true, and that we don't paralyze people that we had no expectation that that was going to happen. And thus, connectomics gives us the ability to make rational cuts in the brain. So for example, if you look at this medial frontal glioma, it allows you to make a cut like this with 3 key networks to remove the tumor in the center. Connectomics also provides the ability to start to think about rehabilitation in a targeted fashion, or a term we call interventional neurorehabilitation. And this really raises the concept of looking at how do we reorganize the connectome for therapeutic gain, particularly leveraging connectomic insights with things like targeted brain stimulation or potentially other therapeutics to improve outcomes. So for those of you who are familiar with TMS, it's often used as a brain mapping technique, but it has a much longer literature as a therapeutic technique when it's delivered repetitively. And what it does is it stimulates a region of brain. So in the pipeline, how do we figure out what's wrong with someone's brain and how we decide where to go? This is an evolving field that we've been able to get research or experience with over the last few years. But essentially, you take the standard atlas here, you personalize it using the reparcellation technique, and then you use that atlas to divide the brain up into subcomponents. You average the bold signal over those subcomponents, and you compare them with everyone to make an adjacency matrices. And then eventually, we use more sophisticated machine learning techniques to begin to compare this to normal individuals and tell us what's normal and abnormal. So again, functional connectivity is comparing the bold signal between 2 areas and getting a correlation between them. And when you do that, you acquire an enormous data set, both structural and functional connectivity. And so this is if you look at the time component, it can be up to a 25 million dimensional data set. You need machine learning and very advanced data techniques to make sense of this. So in this particular matrix, the red areas indicate a comparison between two areas, the small areas on the left being the 379 areas that are mapped in the software, and the same areas on the x-axis. And a red area indicates areas that are highly correlated, and blue areas that are anti-correlated. Now this seems almost intractable until you realize that we've organized this in a non-random fashion. So for example, the reason there are red squares down the diagonal is that these are the comparisons of different parts of different large-scale brain networks. We know this because of our brain mapping research, linking the functions and functional networks, for example, in the supplement and other papers, to areas in the HCP scheme. So we know that these areas, because they're forming networks, are highly correlated, and thus you see red dye in the diagonal. So it's a non-random affiliation. And there's some very interesting insights. So for example, if we look at the language network, these on the X and Y axis are the same list of parcellations, and every square in the matrix is a comparison, with the red being areas that are highly correlated and the blue areas that are not. If we look at a normal one, it's mostly red because the language areas in the resting state are supposed to be firing together pretty often. And on the right side, in this person who has a glioblastoma in the language system with aphasia, we notice that it's more blue because we don't have the synchronization of those areas as much, and they're less functional as a network. So for the first time, we can actually see aphasia and its functional consequences at a functional connectivity level. But more importantly, we don't just have to look at this, we can actually do something about it. So we can decide that, for example, we want to change the functional connectivity of this area by stimulating it. And we can export that to a file. We pick an area, aim it at, select a file, and then stimulate that particular area using image guidance. And again, it's important to note that these technologies are much easier to make sense of functional connectivity problems when you use machine learning to know what's normal and abnormal. So for example, in this comparison of two higher level cognitive control networks, the black areas and white areas are essentially highly variable or normal, which we don't consider abnormalities, but the red areas are areas that are more correlated than normal, and the blue are less than normal. So what we can see is abnormal functional connectivity in a patient treated, scanned on a clinical scanner in a workflow that's accessible to clinical treatment paradigms. Now, the next question about this is this. So we have all this technology. How do we actually use it? And part of this is, and one of my passions is upskilling the community to understand what we're actually getting out of these kind of workflows. And part of this understanding is to really change how we think about brain anatomy, particularly cerebral anatomy. And there's been a lot of interest in white matter over the last several years, or even a decade. And while white matter is important, and I've spent a lot of time with white matter, it's important to know that no one in the history of my practice has ever thanked me for preserving their iPhoth. We care about white matter because we care about function. And we care about white matter that happens to link up networks that we care about that do function. And we know that no matter what kind of surgery you do in the cerebrum, you are cutting white matter somewhere. So we're not, the whole concept of preserving white matter as a means to an end and not as a secondary gain, meaning that you're doing it to achieve another goal, is something that really we need to move past. We need to move towards the idea that we want to save white matter that links different parts of large-scale brain networks, because saving the brain networks means saving brain function. And this allows us to determine better what is expendable and what's not. So when I say brain network, and I've used this term a few times, what do I mean exactly? Well, we know that brain networks are areas that tend to fire together more often than other areas fire together. So for example, as the language system, as I showed, these are two areas that tend to fire together on a common time course and thus have highly correlated bold signals. In our experience, in the majority of cases, nearly all cases, something that constitutes a network has a physical connection. They're wired together. and they often serve a common function. It's also important to know that they actually are not all equal. They're networks that control other networks. So for example, as I'll show you in a second, the language system is actually part of a broader collection of networks called the default mode network. And the default mode network has a core portion, a language portion, an auditory portion, and a memory portion. So how many networks are there? And the question is, well, it depends on how granular you want to get. Now, in general, the best way to think about this is depending on how you set your filter. If you set it at the highest level of granulation, there are about 7 main brain networks. Some of them you're familiar with, like the sensorimotor system. So the motor cortex and the sensory system are highly correlated with each other, as are the SMA. The visual system, which is almost entirely in the occipital lobe. And the limbic system in the lower right. Okay, and this also involves the perilimbic cortices, such as the orbitofrontal cortex. But there are also some networks that you're probably less familiar with, like the central executive network, the default mode network, the Salience network, and the dorsal attention network. And we call these the control networks. Now, it's important to know that any of these networks can often be subdivided further. So for example, while they tend to be more correlated with each other than they are with anybody else, they also can, in some cases, some parts of them are more correlated with other parts than they are with the whole set. So depending on how granular you want to get, you can divide this in up to even, in some cases, 160 or more brain networks. And so a network is really a concept of what areas in general tend to be the major axis of brain function. So showing you through the 7 main brain networks, most people are familiar with the sensorimotor network. Note that the primary auditory cortices are part of that. The visual network, the occipital lobe, and the limbic system and perilimbic cortices. You can see that those are 3 that most people are familiar with. Now, if you look at this orange network, it has multiple parts, and there's a reason for that, and I'll show you why in a bit. But it is basically a network that's involved in active brain function and goal-driven behavior, and called this the Central Executive Network, or CEN. There's a red network, which is mostly anterior cingulate, posterior cingulate, and lateral parietal, though it has other components, as you see, called the default mode network. This pink network is called the Salience network. This is a more recently discovered network. And the dorsal attention network, which is in green. And again, these define the major brain axes, but again, they can be subdivided. Now, how do networks link to function? Well, this is a very complicated question, but in general, there are some brain functions that are mainly linked to one network. So for example, imagination and deep thinking to yourself, theory of mind tend to be a default mode network function. But in other cases, a function is really a specific combination of different parts of different networks mixing together for a short period of time to allow that function to happen. And so when you start talking about the really higher functions like mathematics, or even some aspects of language, these can often be a multi-network function. So again, by linking these to names of places, given that we can now parcellate almost any brain with the reparcellation technique, it allows us, and we've begun to understand a lot about these areas, we can then begin to figure out where in the brain do these areas live by taking all of the task-based fMRI literature, performing a technique called meta-analysis, where we find where in the brain is, for example, the language system in this case, linking them to parcellations by finding out which parcellations or regions lie in those areas, and changing this to a detailed structure of what the network looks like from a white and gray matter standpoint with names. And this is something that we can then scale deeper into the brain of abnormal people to begin to compare. So let's run through a few networks to get people familiar with them. And part of the reason is that we kind of understand what are we trying to accomplish with them? What are these things doing? So the control networks, the DMN and CN being the 2 major ones, sit at the top of hierarchy of other networks. There are other networks whose function or their bold signal correlation are more explained by the DMN or CN than other areas of the brain. And ultimately, they form kind of an axis. And so for those of you who are not familiar with the science on this, essentially, the theory goes as follows. The default mode network is generally on when you're doing internal mental state or not inactive cognitive task. The CN is really more in external goals. And the third network, the Salience network, modulates the switch between them. And so when we began to look at areas like the cingulum, which we thought were a very simple white matter bundle, and we began to map them to parcellations, it becomes clear that these networks are far more, these track bundles are far more linked to the topology of networks, the organization of networks, than we would have thought otherwise. So while we hadn't really noticed that there are all these fibers leaving the posterior cingulate, these fibers are going to this posterior portion of the default mode network and linking them up as follows. So the default mode network is mostly an anterior cingulate, posterior cingulate, and lateral parietal. Now notice that this is the core of the network. There are other parts of the brain, if you remember the photo of the 7 networks, that are not in this core network. And what that means is that they affiliate with the DMN more than they affiliate with the CN, but they're not really fundamentally part of the DMN in its core component. The Salience network is also known as the cingulo-opercular network. It involves a middle cingulate component, an anterior insular component, and the frontal ascendant tract, which links them. And the central executive network is a very complex network, but essentially is 3 parts: the lateral frontal polar region, an area called 8C in the posterior dorsolateral prefrontal cortex, and an area called PFM in the supramarginal gyrus, and they're linked by a string of connections in the superior longitudinal fasciculus. Now, again, I've talked on this briefly, but it's important to note that a lot of cognitive functions can be strongly linked to some of these networks. So the DMN is really a critical part of the brain, and its abnormalities in the DMN are linked to most mental illnesses in various forms. The Salience network is part of coordinating responses to stimuli, emotional regulation, and pain. And dysfunction of the Salience network, for example, it plays a major role in depression, as well as people who have chronic unexplained pain. The central executive network plays roles in executive function and working memory, and the dorsal attention network, which we briefly touched on, is really more involved in direct sustained attention, with a component or a complementary network, the ventral attention network, involved in changing attention between tasks. So again, going over the networks again, you have the sensorimotor, vision, the limbic. You have the CN, the default mode, the salience, and the dorsal attention. Now, when we start to look at combinations of networks, we can start to track important clinical syndromes. So one of the ones that we've been working on for many years is what happens in the medial frontal lobe when patients become akinetic mutes. If you look at the anatomy of how the default mode network and the salience network talk to each other, and the fact that the salience network shares a component with the SMA, it becomes quite a lot easier to understand how the SMA syndrome and the salience network are linked to each other. And this has been also confirmed by a group at Harvard who demonstrated that strokes, particularly in that little band, specifically cause damage to free will. And we've called this the initiation axis, which we could consider now an eloquent part of the brain. Now, it doesn't mean that no one can ever remove a tumor or anything like this from here. Just like there are tumors removed from the motor cortex, anything can be removed if the brain's reorganized or the patient can tolerate it. We're still learning about how important this is in certain contexts, but we do know that this is probably the mechanism of how people get this kind of problem, and that it's possible that even a unilateral singulectomy in this region can be damaging to patients. Moving on to a brief discussion of language, and again, this could be a talk by itself, but we were taught this model in medical school and residency, but it doesn't really explain a whole lot about what we see about particularly in the higher aspects of language. One of the things that it fails in is the concept of semantics. So for example, people have noted that semantic dementia, which is a type of frontotemporal dementia where people have fairly fluent speech but a general absence of content behind their speech, involves the left temporal pole and doesn't involve any of the classical language areas, raising the idea that there's a concept of 2 pathway models of language. And you can see 3 here that involve various combinations of either the ILF, the inferior frontal occipital fasciculus, or a collection of fiber bundles along the middle temporal gyrus. So when we began looking at this, obviously the first thing we looked at is where's the core of the language network? And these are the regions that we found. Interestingly, this arrow here points to an area called 55B, which again sits in the back of the middle frontal gyrus in the precentral sulcus, an area that we didn't think about as a language area. But there's a lot of evidence, including a recent Cell paper and a case report that was published in Neurosurgery, that we performed with Eddie Chang at UCSF, that suggests that this is probably a motor cortex for the larynx. And this is the case that we reported. Basically, you can see it's a relatively modest resection of a glioma in this area, and this patient has fairly normal speech, language content, but is really unable to phonate, has a pure apraxia of speech, which again, we would have never thought about this as a speech area until we understood the anatomy of this. In fact, when it happened, there were years before we understood what happened to this patient, but now we had a better understanding. Next question is, should we really be calling things speech mapping or language mapping when those are really two different functions that are related but not the same? Speech is a motor function, and language is really more of a cognitive function overlying the motor function of speech. And so when we began to look at how does the information leave the temporal lobe, the back of the temporal lobe, and how does that information get to where it's going, and what does the temporal pole have to do with it, we found this really interesting diagram, which is from our supplement, and it's looking at the functional connectivity of an area called left TGV, which is, again, the area linked to semantic dementia. The areas in yellow are areas that correlate with it. So you know that there's a band between the posterior left temporal lobe and TGV on the left. What's also interesting is that you see the Broca's area and area 45 in the left inferior operculum that are also lit up. But finally, you see the classic pattern, which hopefully you recognize now, of posterior cingulate, anterior cingulate, and lateral parietal of the default mode network, which suggests that this higher level of function is involved in a more verbal memory and semantic processing stream that is essential for higher aspects of language. And this may provide some anatomic understanding of how patients who undergo temporal lobectomy or even selective nuclear capectomy develop some of these language disturbances that have been documented. But again, this is complex. There's also the ILF pathway. You can see this basically comes from some of the visual naming sites to the temporal pole. And furthermore, we found that there's an anatomy of this temporal stream, which involves a series of complex U fibers that parallel the middle temporal gyrus, again showing that there are multiple pathways that are necessary to integrate a visual and verbal recognition that eventually extends to the speech areas, probably via the uncinate fasciculus. So again, in addition to the classic Broca-Wernicke type arcuate model, we have areas that go to 55B. We have the parietal regions, which are probably Geschwin's area. Notice that these are also part of other networks. We have the default mode network component and the accessory language pathway. We have the ILF component. We even have a direct connection between the auditory system and Broca's area that's independent of all of these. And ultimately, this leads to a much more sophisticated modern language model, which is multifactorial, multi-component. And again, this could be a talk in and of itself. Now, as we run through additional networks, so for example, the visual system, again, what you can see is that we have a lot to study on what are all these pathways doing? What's important? How do we preserve higher aspects of vision? And also, why does the brain dedicate a significant amount of its white matter to connecting very higher level areas like frontal polar regions to the earliest visual system like V1, which is a very significant amount of the white matter of the brain? So data that we have from schizophrenia suggests strongly that pathways like the IFOF are predominantly cognitive pathways, and the dysfunction of these pathways lead to cognitive disturbances as much as anything. And this really arises from the fact that the visual system it requires a lot of top-down processing to regulate what's salient, what's important, and how the visual system is processed. And again, if the visual system processed all the information that was presented, the actual visual system will be bigger than the brain. So we know that top-down regulation is necessary to focus on what is important and what's not, and to regulate the visual pathway processing. And again, this is a significant role that ILF and IFOF are likely providing, showing how they may actually be still playing a role in different aspects of visual recognition, in ways that we don't totally understand yet. Also, Connectomics allows us to tackle things like apraxia. You can see that this is a fairly complicated system, but mainly is focused around the left parietal sulcus. And it seems pretty obvious why most strokes that cause aphasia would cause apraxia, but that this is possibly dissociable from language problems. Finally, a brief discussion about neglect. Now, where is neglect located in the brain? And again, we know that it's the right parietal lobe somewhere, but that's not very helpful when we're trying to preserve it in surgery. So we did a little bit of a deep dive and performed meta-analysis, and you can see these red blobs are showing you where in the brain are lesions that across studies statistically are associated with neglect. Now, for those of you who are familiar with white matter anatomy, you probably recognize that this looks like the SLF2 and arcuate complex. And this is predominantly a white matter problem. So we began tracing those white matter pathways to see, well, what were they connecting? And what you can see is this collection of parcellations. Now, I know most people probably don't look at this and can't recognize this, but we've labeled them for you. So there's components of the auditory system, the default mode network, the olfactory system, the dorsal attention network, the somatosensory system, and the frontal eye field. And so at first we were kind of baffled because it didn't really fit with what we expected. But what we eventually figured out is that neglect was a problem that occurred when the fibers that joined different parts of different networks that were allowing them to make a combined version of a single world were damaged. It's really a problem of white matter. And we also addressed the possibility that this was either the dorsal attention network or the ventral attention network. The ventral attention network is particularly appealing because, again, it's a right-sided only network, so we thought this thing seems too good to be true. But the data don't line up with that. The data suggests that while there's parts of both of these networks inside of the neglect system, neglect is its own system of white matter fibers that join networks that do slightly different things. And it highlights how function is a mixture of different parts of different networks. So neglect is not a place, it's a white matter connections that allow different networks to talk to each other. And this highlights the fact that things like mathematics involve multiple parts of multiple networks and a common core that link them, and really changes how we think about cognition. So there isn't a math part of the brain, there's a math network that allows different parts of different networks to talk to each other. So going back to the main networks, so what do we do with this information? Well, it's safe to say that cutting across the middle of major networks is going to take a lot of cognitive functions out at once. It may not be possible to preserve all parts of every part of the brain, particularly with some very bad brain tumors, but it seems reasonable to say that cutting across the middle of a major network is going to do more damage than any other situation, because if the mixing of networks is necessary to create cognitive functions, then if the networks can't talk to each other, how do we expect cognitive function to emerge out of that? But this seems, at a first glance, to be an impossibly tractable problem. How do you memorize a map like this? And the answer is a lot simpler than you think when you understand the logic. So again, everyone are familiar with these three networks, so I won't go into it. But what about the other networks, the four control networks? CN, DMN, Salience, and dorsal attention. Well, it turns out that there's a complete logic to this that runs deep throughout the nervous system and is striking once you see it. And once I show this to you, you probably can't unsee it. So let's look at the medial frontal lobe. Notice that there's this repetitive pattern of how those intertwine with each other, these V-shaped structures in the medial frontal lobe. And further, there's a pattern, this pattern is continued throughout the brain in a repetitive series of subunits. So dorsal, the default mode network, follows this sort of S-shaped pattern. It goes from posterior to anterior, comes onto the lateral surface, goes down to towards the insula, back towards the parietal lobe and the inferior parietal lobule, and finishes at the lateral temporal lobe. As you can see, those last three parts are the language system, making a kind of an S. Oh, it's CN, starts posterior, has that V structure, comes onto the lateral surface, heads down towards the insula, goes back to the inferior parietal lobule near the interparietal sulcus, and then goes down to the temporal lobe. Same pattern. And the salience network starts in the back, has this V, heads down towards the insula. Actually, it's this main component. Heads back to the inferior parietal lobule, and has a smaller portion in the temporal lobe, making this S. And the more primitive dorsal attention network is really an incomplete S. So this repetition is striking, and it isn't limited to the neocortex. So when people began to look at the cerebellum and the functional connectivity of the cerebellum in a classic paper, they found that when you looked at it in this coronal view, it wasn't very informative. But if you looked at it in the sagittal view, this began to become very logical. So notice that when we look at the cerebellum in the sagittal view, starting from the very top, so roughly about 1 o'clock on the clock, you have the motor system, the salience network, a small portion of the dorsal attention network in green. The O is the CEN. Then you have the DMN on the farthest portion. It flips back over the DMN again, the CEN, dorsal attention, salience, and sensorimotor, showing that inside the cerebellum, the majority of the cerebellum is not motor, it's involved in managing the control networks. This is also repeated inside of the caudate and putamen, where you see that there's clear segregation and that segregation strongly follows the same basic sets of patterns. So the most rostral portion of the caudate head and caudate tail is a default mode network, then eventually CEN, salience network, and sensorimotor. And this shows that there's almost certainly a developmental pattern in these networks that is of repetitive subunits that surround the whole brain. So why are the control networks all over the brain? Well, it's fairly simple, and there's a logical explanation for this. So we think about motor systems, motor planning systems, like the supplementary motor area, as a motor area. And that's not entirely true. So if you look at this map here, which shows the different parcellations of the HCP scheme in different colors based on what network there are, it becomes clear that the supplementary motor area, which has 4 parts, has a portion of salience network here in orange, this area in green called the superior frontal language area, and a blue area that's part of the sensorimotor system. So again, a mixture of different networks in the SMA. This is repeated in the dorsal premotor area, which is a mixture of motor, dorsal attention network, and salience network in one area. If we look around Broca's area, an area many people are familiar with, we notice that it's actually a combined area. There's part of the default mode network, that's the primary canonical area 44. There's part of the central executive network right there. There's the dorsal attention network, there's a salience component, and a motor component, all linking around the motor area to the face. And so what we believe you're seeing is that the reason the control networks are in different parts of the brain, little components intermixed with each other, is they allow different cognitive components to rule in on how the brain works in different ways and put them in the position of different parts of the brain at different times. And this is how through evolution, as the brain evolved, how it continued to allow higher and higher systems to maintain control of lower systems to drive them towards cognitively relevant tasks. So now that we've kind of upskilled our understanding of connectomics and the anatomy of brain networks, what do we do about it? And a lot of people have asked me, how do we use this? I finished writing a book about this. I think it covers this in a fair bit of detail. And what I'll talk about, what really is in that book, and kind of my thinking on this topic, I learned that supratentorial brain surgery was basically the following algorithm. You determine whether something was eloquent or non-eloquent. You generally wanted to stay out of the eloquent areas, stay in the tumor, and don't cut the corticospinal tract. There are other things, but in general, this is basically what you're trying to do. But the problem with this is, while this is reasonably effective, everyone who's being honest knows that there are neurological deficits we can't explain. There's sometimes that people have language problems, like the case I showed earlier, and we don't know why they have them. And more important is that many people do not go back to their pre-surgical baseline, even when we've had a successful surgery. And part of it is we don't know exactly what we're doing wrong in these cases. But worse, without data, we don't have a way to iteratively improve and to learn from our mistakes. We just kind of shrug and say, well, the frontal lobe is mysterious. Connectomics is the first real, in my opinion, concept that allows us to realistically begin to look into the black box and learn from our mistakes. And whether we admit it or not, we are cutting next to brain networks. Just because you don't know that they're there, just because it's not on your image guidance, does not mean that they're not there. And the subcortical anatomy is anatomy. Now, there's been a discussion about calling all brain eloquent. And there's papers that have been written about this. Now, it is without doubt that all of the brain is active. It didn't evolve to suck CSF. It didn't evolve as a cushion for the motor cortex. So it's really insane to think that we can just go hog wild in the frontal lobe and nothing occurs as a result of that. Having said that, we know that there are parts of the brain that you can cut that are worse than others, and some areas that are fairly well tolerated. So what the real challenge there is not that all parts of the brain are eloquent or that there are parts of the brain that are not eloquent. It's that we're not making any of these decisions based on facts. And even though we don't know it, it's there. So when people ask me, what is the utility of DTI? DTI is not going to protect, or anything else, Connectomics is not going to protect you from doing something stupid on itself. The best way to think about its utility is to think about it in terms of the Venetian lagoon. So for those of you who are not familiar with the Venetian lagoon, it's an area of relatively complex waterway between the Italian mainland and the city of Venice. Now, he protected Venice for centuries, and Venice maintained its independence till 1811 from the Middle Ages, not because people didn't try to take it over, they tried to take it over all the time, but because of the Venetian lagoon, if you didn't know how to navigate it, you would often wreck your boat. It would end the invasion before it started. Now, when you go to Venice now, you see that there's a series of posts that tell the water taxis where to go. And so in this analogy, the Venetian lagoon is the white matter. It's amorphous, and there are a lot of traps. The posts give you a framework to try to navigate this. Now, if you want, you can wreck into the post. If you ignore the post, or if you just get unlucky, it's still possible to wreck your boat. But at least you're not going to do this because you don't have a mental framework of what you're trying to accomplish. So DTI allows us to understand the framework of the subcortical space and to figure out, how do we separate this from the tumor? What do we care about? And how to make goals and plans for surgery based on facts. In our book, I talk about several disconnections. Now, it doesn't mean that every single case, a temporal lobectomy is appropriate. Sometimes it is, sometimes it's not, depending on the situation. What it just says is that these are cuts that generally respect the anatomy of the brain networks. And if you need to do a temporal lobectomy or a frontal lobectomy or an insular resection, this is what you're basically trying to accomplish. And it gives you some corridors that are reasonably well tolerated. Not that none of these have consequences. There's consequences to anything you do in the brain potentially. And we still don't have tools that tell us when the anatomy is not the same in the situation. But this is a first guide towards being able to actually respect at least the core of what we think are major brain networks. So for example, this medial frontal disconnection respects the frontal axial tract, which is in red, and that's an important part of the Salience network. It respects the cingulum, which is an important part of the default mode network and Salience network, and respects the arcuate fasciculus and SLF, which are part of lots of different areas of the brain. Now, in order to really leverage this and to bring those kind of connections into reality, what I found as I began to work with people on this is that it was really asking a lot to expect everyone to be able to reconstruct this on their own. And so we worked on software that we call Quicktome to make this very simple. And what Quicktome basically does is it essentially limits the workflow towards selecting montages of networks that are generally relevant to the surgery being performed. So for example, if you were to click medial frontal and click what side you're on, it would generate a set of objects like you would here, look at here. And while you would still have to look at this and edit them potentially if they're incorrect, it utilizes machine learning and heavy level cloud pre-processing to basically simplify this to where you're basically back to anatomy and things that you're familiar with without having to be able to do all the coding and pre-processing yourself. And it does this in a clinically relevant timeline. The goal eventually is to get this onto image guidance so that you actually have this information at surgery. And there are a lot of things that we had to use technologically to make this usable, because again, as a neurosurgeon, I know that we don't want to waste people's time or make this more complicated than necessary. So for example, doing a lot of noise correction on the front end, utilizing tractography that aligns with the anatomy. So for example, CSD, constrained spherical deconvolution, which is better at showing crossing fibers, and utilizing workflows that basically work the individual out of the workflow as much as possible. So the ability to basically push the scan directly from the scanner into the cloud, have the processing done, and then just focus on editing and exporting your target selection, which again, eliminates the need to be a computer expert to do any of this. So once we have the ability to create montages and link them to specific disconnections, so for example, this would be the medial frontal disconnection I showed a second ago, we can then begin to utilize this to make safe cuts in the brain. Now, again, you may not necessarily do a frontal lobectomy here, but again, what it comes down to is if you needed to remove the entire frontal lobe, this would be a way that would preserve as much of the anatomy as possible. And so this is an example where you do need to remove the majority of the frontal lobe. You can see that this is a pretty extensive frontal tumor. While we might be able to preserve that front anterior cortex, this is probably not very important, given the fact that remove this tumor, you're likely going to disconnect that from the rest of the brain. And so in a glioma, if it's going to have cancer in it and it doesn't talk to the rest of the brain, you might as well get the oncologic benefit of removing it. Now, what are we trying to accomplish in this surgery? Well, you could go in there and suck the tumor out, but you know, again, it's sort of like playing chicken, where eventually you might hit the ACA or something like this. And again, it's often better to work on the back of the tumor where the danger is from the beginning, so that when you get to the removing this large mass, you can do it in more of a non-block fashion. So if you look through this workflow, what you can see, for example, is there's three main networks at risk. The sensorimotor system, which has a kind of a more familiar coronal cut that preserves the connections between the motor planning areas and the motor system. The default mode, which as you can see, the Quicktome has been able to map the parcellations of this, even though they're up against the tumor. Now, what's interesting is, while those look fairly compromised, and it may not be possible to save this, keeping that medial portion connected posteriorly, if possible, is a good goal because it actually keeps the network connected to itself. So this is why understanding network anatomy is very important. And then, of course, here we have the central executive network. Now, part of that's in the tumor, according to this, but some of it's not. And so what this allows you to do is kind of make an understanding of the oncofunctional considerations to preserve as much of the CEN as possible, but still do an aggressive tumor resection like we did here. And notice that, again, we knew that the portions that were in the deep basal ganglia, and we decided not to go after that and treat it with radiotherapy with good results. This patient's been functional for quite a while, even with a very good aggressive tumor resection. Looking at two other connections quickly, in the temporal resection, the main anatomic consideration is that you're dealing with three systems: a lateral system, which has either neglect or language pathways in it, the medial system, which is fornix and hippocampus, and the inferior system in green, which is the ILF. And what your goal really is to get past the red and into the blue if you're dealing with something like a hippocampal glioma. Because, again, those systems are generally not crossing the ventricle and talking to each other very much. So the main goal of the resection is to work underneath the language system as much as possible to get into the hippocampus. Now, sometimes you're doing this as part of a temporoinsular resection, and you can see the Quicktome outputs look like this. And there's a couple considerations. First is the temporal lobe will have to come out. You'll need to remove a portion of the anterior temporal pole, and you can see in the neglect system on the lower side. You can see that the IFOF is on the tumor side is relatively atretic and pushed medially. So you want to try to save as much of that as possible, but on the assumption that some of this may be reorganized. And on the far left, you can see that the face motor fibers, which are, as you can see, are very well visualized in CSD because it's able to handle that crossing fiber, are draped over the top of the tumor. So while you are unlikely to injure those directly doing the insula portion, if you escape the insula, the corticospinal tract is always nearby. So again, the cuts with the corticospinal tract look sort of like this, and the cut with the neglect system looks like this, entering the ventricle and working underneath it. And again, there's a fairly good opercular portion here that also needs to be preserved. Finally, looking at a medial parietal disconnection, you can see that in this very large butterfly tumor, that there's a lot at risk. And so now we kind of can get an understanding of why these tumors can be so bad. So not only is it impacting the sensorimotor system, that large cyst, and it's up against the language system laterally, but it's wrapped around the default mode network. And this is something that we never really thought of before, and we never could see it. But as you can see, that with edema correction and artificial intelligence, it's possible to see the tumor and exactly how it's impacting this. Now, whether or not you could actually both control this tumor and remove it and save the DMN is a very challenging question, and probably in some ways there's going to have to be some kind of compromise here. But it also shows you why these patients have such profound memory and cognitive problems, because this is the nature of this tumor. So if you're attempting to tackle it, notice that there's several cuts that you're really trying to make with different brain networks. And this is challenging, and it may not be a case that you can achieve an excellent outcome with. But what the fact of the matter is, it shows you what you're really there to accomplish and what really is compromised in this very complex case. And again, if you were going to go try to attempt to at least debulk some of this, you now have a clear guideline on what you would be doing. So finally, what do we mean by disconnection? So this video kind of walks through this a little bit. So you can see this is a medial parietal disconnection. We've mapped for neglect. We found areas that cause failure of target cancellation. And what we want to do is to make a cut kind of along where I'm tracing with the bipolars, and to basically preserve the superior longitudinal fasciculus and portions of the dorsal attention network as possible, getting down to the ventricle to remove a very large tumor that's extending down in that direction. Now notice that, of course, the challenge in this operation is we don't see any of these pathways. Image guidance isn't going to be millimetrically precise. So even if the tractography was perfect, it's only a guidepost. But what we really have here is now we have a clear idea of what we're attempting to accomplish. So you can see here, this is roughly where the SLF is. And what I'm trying to do is continue this cut, preserving that plane to go deeper and deeper until we get down to the ventricle, at which point we've cleared the pathway as much as we can. And again, through this resection, we're monitoring the patient throughout the disconnection. And what we now have is an anatomical understanding of the subcortical space that allows us to make the best decisions possible, given how terrible this tumor really is. And while there may be additional consequences for what the damage this tumor did or this surgery, this is something that we can iteratively improve on over time to, again, continue to get better at this. So thanks everyone for listening. And if you have any questions, feel free to contact me. I'm always happy to walk through these kind of concepts with everyone.